Thank you so much for that introduction. So, as I told you before, my name is Anthony. I'm from Peru. I belong to the molecular biology lab here in the National University of San Marcos in Lima, Peru. And with my team, we are going to present us this topic, some of our latest investigations. This time, I'm going to talk to you about the characterization of the Pictolysin-I, a novel P1 metalloproteinase isolated from Botrus pictus venom with antitumoral and mitochondriotropic effect on triple negative breast cancer cells. So let's do it. So first of all, it's worth remembering what Botrus pictus is. Described by this guy, Johann Jacob von Schuri in 1845, Botrus pictus, commonly referred to as the desert landscape, is a pit viper endemic to Peru, which is distributed in the western slopes of the Andes, the coastal Foji river valleys along the central to northern Pacific coast here in Peru. And in this part, Botrus pictus is responsible for the major cases of snake envenomation. As you can see, these are some of the symptoms of envenomation. And this is because of its venom composition. That is why it's so important to understand how Botrus pictus venom composition is. So first of all, well, we have to remember that Botrus pictus is a Viperidae species. So as you can see here in this diagram, Viperidae snakes has this type of protein represented in green, which is the metalloproteinase, snake venom metalloproteinase. And these are the major component of the venom, as you can see here, but also the phospholipase, A2, and so on. In comparison to Elapidae, Viperidae has more metalloproteinases in its composition. And when we talk about the Botrus genus, the Botrus, in this case, the genus of this species, Botrus pictus, we can see here that the composition of the metalloproteases is, is major. It's the most representative toxin in its composition. And particularly in Botrus pictus venom, as you can see here in this another diagram, we can find two types of metalloproteases, the type 3 and the type 1. Being the type 3, the major component, more than a half, and the next metalloproteins is the type 1, the P1. So in order to understand the importance of these metalloproteases, we're going to see the next slide. So why are these toxins so important for snake envenomation? These toxins are so important because they are the primary factors of hemorrhage. That is why it's so important to study this toxin. What is more, these are the most abundant toxins in the venom composition. So we need to understand its mechanism of action and the types that are in the venom. So as you can see here, there are three types: P1, P2, and P3. P3 is the more complex. As you can see here, it's P3 because they have three domains: the catalytic domain, the disintegrating or disintegrating-like domain, and the cysteine-rich domain. This is another one which we can find in some species, like this one, the Boya siamensis, but only that species, not in case of Botrus pictus. But in general, we can find these three domains: the disintegrating, cysteine-rich, and the catalytic domain. In case of Botrys pictus venom, we can find type 3 and type 1. And in this case, I'm going to talk you about the type 1, which is only the catalytic domain without the system reach and without the disintegrating domain, only this domain. But also we can find in each one the peptide signal and the prodomain, which after processing, it is excluded of the toxin, the major toxin. So how we can have this P1 toxin? In our case, we use size exchange chromatography. These techniques permit us to separate to fraction the proteins of a mix. In this case, the mix was the venom. We use the venom, we introduce two columns, and we use this type of resin, cefacryl S200. And here you have the chromatograph. We can see three peaks. This one which is a little peak, and this the last one. But also, we perform some biochemical assays like azocasinolytic assay, but also BAFNA assay. Azocasinolytic permit us to make a screening of metalloproteinase. BAFNA permit us to screen in this case, the serine proteins. So as you can see here, this graph, the orange line, represents two peaks. This one is the P3, and this one is the P1, the protein of our interest. And here we can see also another peak of BAFNA, which represents the serinoproteases. Here we have an SDS page, and here is the P1, which I'm going to show you more in the next slides. So this was the first step of purification of science exchange chromatography. The next one, and the last one, was Cefadex G50. We pulled these fractions and we introduced to a different resin in order to separate these proteins. So we separate, we perform the azocasinolytic activity as well. We recognize this peak of activity, the orange one, which represents the peak one, the Pictolysin-I. And you can see that protein as well here. But in the next slide, what we can find is the purification table, where I'm going to show you, in this case, the amount of protein that we introduced to the column, in this case, 82.7 milligrams, and then the purification performance, the total enzymatic activity, and so on. As you can see here, the purification performance decrease because in each step we lost protein, that is why. But we also can see the specific activity, how it is increasing because of the purity of the protein, in this case of the Pictolysin-I. And here we can see the SDS page in order to confirm the purity of the protein. Here is my protein, Pictolysin-I. It weighs like 22 kilodaltons approximately. And here you can see the steps. The first one is venom under reducing conditions with beta-mercaptolethone. This one is without reducing conditions, as well as venom. This one is cefacryl S200. In this case, reducing condition, non-reducing. And this one, the last step, is Cefadex G50. Reducing and non-reducing condition. And here the ladders, of course. So we can see that the protein, in this case, Pictolysin-I, weighs less than 25 kilodaltons in this case, and it's approximately 22 kilodaltons, its weight. So once we isolate this protein, this toxin, we perform the biochemical characterization. So regarding this characterization, the first activity that I performed was the effect of ions and inhibitors. So as you can see in this graph, we can see the residual activity, in this case, azocasinolytic activity, and the effect of the different ions like magnesium, zinc, calcium, and different inhibitors like DTT, EDTA, and beta-mercaptoethanol. As you can see here, there's a significant decreasing of the activity. And in the case of the ions, especially the calcium enhance the activity. And it is significant, as you can see here. Regarding the effect of the pH, we can see here that in this case, 9 pH or pH of 9 can increase significantly the activity, the proteolytic activity, and a pH of 10 as well, as you can see here. But in case of a more acidic pH, we can see a decrease of the activity as well, as you can see here. And regarding the temperature effect, we can see that in case of the temperature of 5 Celsius, there's an increasing of the activity. But if we increase that temperature, like in this case in 75 Celsius, we can see a decreasing, significantly decreasing of the activity. So we can say that the temperature, the ideal temperature optimal could be in this range between 5 Celsius, 5 degrees Celsius, or 25, preferably. And regarding the effect of the antivenom, we can see a neutralization of the activity, but in comparison to the control, we can see an increasing of the activity despite we use the antivenom. In this case as well, when we use the antivenom with the venom of Botrus pictus, we can see an increasing. This result is probably because of the formation of immunocomplexes. As you can see here, without Pictolysin-I, without venom, when I perform, in this case, the azocasinolytic activity, if I use more antivenom, there was an increasing of the absorbance. And that is why, probably because of the immunocomplexes formation. Regarding the biological characterization, we perform a hemorrhagic activity. In this case, using Botrus pictus venom as a positive control, Pictolysin-I, in this case, 16 micrograms, and PBS as a control as well. In this case, in case of PBS, we cannot see the halo of hemorrhage. But in case of the venom, of course, we can see the halo in these cases. But when we use Pictolysin-I with this amount of the protein, we cannot see this halo, this effect. So maybe we can say that it's a non-hemorrhagic toxin or non-hemorrhagic metalloproteins. This SDAS page represents the proteolytic activity against fibrinogen and fibrin. In this case, we can see the fibrinogen and its 3 chains: alpha change, beta change, and gamma change. And these are treatments with Pictolysin in different times, with different quantity of hours, and incubation, of course. And as we can see here, when we finish the 50 minutes, we can see that the alpha change disappear. In the case of 30 minutes, of course, disappear as well. And then, but also we can see the presence of different, in this case, parts or fragments of proteins that were cleavage because of the Pictolysin-I. So it has an activity in the alpha change mainly. In case of the fibrin, the proteolytic activity on fibrin, we can see here the gamma dimer, the alpha beta change, but there's no an effect in all of the cases, as we can see here. Despite the different types of incubation, we cannot see an effect, a significant effect of degradation. So we also perform a molecular characterization. And to do this, we make a primer design, as you can see here. This is the formal primer plus prodomain, because we want to sequence also the prodomain. This one is the primer without the prodomain, and this one is the reverse primer, as you can see here. We amplify the gene using PCR, and we sequence the amplicons so we can get this sequence. This is the full sequence, including the peptide signal, signal peptide, but also the prodomain and the domain of the metalloproteins, of course. And its length is of 1,230 base pairs, as you can see here.
You've got 3 minutes left, Anthony.
Oh my God. Here we have the amino acid sequences, which obtained using a server, in this case, a bioinformatics server, which is the ORF finder. Here we have the signal peptide, the prodomain, as I told you, and the metalloproteinase domain. Here we got the electrophoresis, and we can see here, this one is Pictolysin-I plus the prodomain. Here is the prodomain, the 3D structure, which performed using alpha-fold 3. Here's the peptosignal. Here's the mature protein, which we can see here as well. We perform a multiple align using different sequences of metalloproteins P1. But the most interesting thing is this part. We can see that the canonical zinc binding motif is conserved, rather the sequences. But also we can see the omega loop, which is important for its activity as well. And here, there's a mutation region, which was modified by Camacho and his team. And I'm going to explain you about that afterwards. This one is the phylogeny. We can see here that Botrus pictus is not closely related to Botrus. It is isolated. But also we can see here the measure protein. This is the omega loop, which is so important for the activity. Here, Camacho made a modification of this, in this case, of this loop, and it removes the hemorrhage activity. And that is so important. I'm going to explain you why afterwards. And this is the canonical zinc binding motif with the zinc, of course. Well, finally, regarding the effect on cancer cells, we can see these three essays. Sorry, I'm going to keep this. But sorry. Okay, wait a minute, please. I got
25 seconds.
Okay.
Tick.
I'm
Tock.
about to finish. Okay, this is
You get a few bonus minutes, but try to wrap it up.
Okay. So regarding the effect on cancer cells, we got these three experiments. First of all, the NTT assay. As you can see here, we use Pictolysin. In this case, we incubate in MDA MB231 cancer cell lines, which is the cell line of triple negative breast cancer cells. And as you can see here, there's a decrease of the population, which is a little significant, as you can see here statistically. But also in case of the normal cell line, not the tumoral, in this case MCF10A, we can see an increasing of the population, and this is so particularly of this protein. Regarding the membrane potential effect, we can see here that there was a little increase of the mitochondrial membrane potential, and that is why this is because of the effect on the mitochondrial metabolism of this protein, because it stimulates the production of ATP and so on. But this occurs in the tumoral cell line, but not in the case of the normal cell line. And finally, regarding the mitochondrial ROS production, as we can see here, there's a slight increase of that production, but it is not significant. But in case of the control, yes, we can see the increasing using Menadian. Well, that's all for me. These are my biographic references. These are the acknowledgements. Thank you to my professor, my beautiful team lab, my assessors, and my friend Daniel Torrejon. Thank you so much.